Good afternoon, everyone. Again, welcome to this webinar from Jasco. Today, our colleague Forrest Cole is going to be talking about orthogonal similarity assessment of monoclonal antibodies using circular dichroism, FTIR, and Raman. A little bit of introduction of Forrest. Dr. Forrest Cole is an application scientist here at Jasco, specializing in electronic spectroscopy, such as circular dichroism. Forrest received his PhD from Ohio State University, where he studied eumelanin, which is a heterogeneous carbonaceous material using FTIR, UVVs, and ultrafast transient electronic spectroscopy. He also studied DNA bound to silver ions using transient IR and UV visible measurements, CD spectroscopy, and other spectroscopy techniques. So with that, I leave you with Dr. Forrest Cole.
Thank you for that introduction, Carlos. So like Carlos said, today we're going to be talking about similarity assessment of monoclonal antibodies, particularly using circular dichroism, Fourier transform infrared spectroscopy, and Raman. And we're really going to be talking about this also in context of a new piece of software that we've developed to sort of bridge a bunch of different spectroscopic methods together and analyze spectra in order to actually make similarity assessments. The software in question being qHOS, which stands for Quality of Higher Order Structure. But before I sort of get into the actual analysis itself, I want to give some background as to what is this qHOS program doing. In particular, why did we go out of our way to develop a new software package? What are we trying to do, or what problems are we trying to solve? And that sort of story really starts off with talking about therapeutic antibodies. So if you're familiar with antibodies, over the past decade in particular, the market for antibody production in terms of therapeutics has basically exploded. It's become very, very high in growth. And this journey of developing antibodies, in particular monoclonal antibodies for therapeutic purposes, really started in the 1970s with the development of the first mAbs, as I'll start to call them now instead of monoclonal antibodies. And with the first development of mAb materials, they became sort of prime candidates for very specific drugs with very specific types of targets, right? So monoclonal or antibodies in the body are already doing that sort of work, right? And so why not utilize those properties for drug development as well? And so the first therapeutic antibody to really get on the market was what's known as rituximab, which is an anti-CD20 aimed at B cells and lymphoma. And then from there, just more and more antibodies started to get accepted as therapeutics, and development really ramped up, developing either mAbs for new targets, for things such as melanoma, bone loss, a variety of different cancers. And then you also had development of materials that were going after the same targets as other mAbs that were developed along this pathway. And with this growth in obviously the production of these monoclonal antibody, these mAb materials, there was also a growth in ways to study these, right? So you want to look at these materials in a variety of different ways to establish that, one, you have good material, and then two, that material is either long lasting or stable enough for use, storage, and transportation. In particular, circular dichroism is one of those techniques that have been primarily used to study the structure of these proteins. And to get into how that analysis really helps analyze monoclonal antibodies, I'll first go to this monoclonal antibody that I've already mentioned. So this rituximab, mainly because it's been on the market for so long, and then it's also very well understood at this point relative to maybe some newer types of antibodies that have been developed. So like I mentioned, rituximab, the way that it operates is that it binds to what's known as the CD20 protein, which exists in the membrane of B cells. And this interaction between the antibody and the B cell is really what helps it effectively fight, in this case, non-Hodgkin's lymphoma. And in order for this interaction to really take place, the structure of the antibody really needs to be correct in order to have effective binding to whatever its target site is. And so we really need to make sure that the heavy and light chains are what we think they are. And we really need to make sure that if we have done any sort of manipulation where we have maybe human region or purine region in our monoclonal antibody, we want to make sure that everything is structurally how we think it should be. And so in this case, let's imagine that this antibody is one that's been on the market a while. We know that it works well, that it's structurally stable. And the question is, if we now maybe have developed a new method of formulation or of producing these antibodies, how do we know that they are actually structurally similar? And there's really a variety of ways that we could go about doing this. And so legislative bodies and things like the ICH, so the International Council or Conference of Harmonization, and of course the FDA, and the European Pharmacopoeia and USP, the United States Pharmacopoeia, have also really thought about, well, how do you determine whether things are different between two monoclonal antibody structures or just biotherapeutics in general? And we can ask this question in terms of CD spectroscopy. So in the bottom right here, we have two spectra, one in the red, which belongs to what I'll call our innovator product, which is basically a product that has existed on the market a while, it's well established. And then we can have now this blue-tipped monoclonal antibody, which I'll call our biosimilar, which is basically the common language used to describe these types of derived materials. Now, one of the things we want to do if we're making basically a product to match what's already on the market is we want to know, are these structures statistically different? And is that going to possibly harm its ability to bind or operate effectively? And in terms of CD spectroscopy, IR, things like Raman or even UV visible fluorescence, we can then boil that question down to, are there significant differences in the spectra? So if you're familiar with circular dichroism, you know that the CD spectrum is a really good reflection of the structure. And so if you know that there's differences in the spectrum, you can verify possible differences in structure. But the question becomes, how do we actually know that there are differences? So we can look at this visually and see, well, there's some mismatch there. But the question becomes, is that statistically different? And what are the safe limits that the councils like the FDA and the ICH can define? In particular, I want to draw attention to a sentence that's pulled out of the most recent FDA guidelines on biosimilars. And their specific statement is that emphasis should be placed on developing orthogonal quantitative methods to definitively identify any differences in product attributes. And I want to put particular emphasis on orthogonal and then quantitative. So basically what they're stating here is that, sure, you can show that a spectrum is maybe visually different, but there needs to be some quantitative measure that's easily reportable and easily verifiable by whatever body is overseeing this area. So in particular here, we're talking about medicine. And then orthogonal means, sure, one number is nice to verify that the products are maybe similar or different, but really you need a multitude of data types in order to definitively say whether two things are the same or whether two things are different. And so with this in mind, we really went in to develop qHOS in order to bridge a variety of spectroscopic methods that are used commonly to study protein structure. And we wanted to distill each of those methods into an easy to understand, basically statistical package that you can really just take the spectra you've collected, put them through qHOS, and then you get back out both visual information that's easy to digest, and you get quantitative metrics on similarity. And so qHOS, what it really is it's a statistical similarity assessment. So it's going to define whether two materials are the same. And then it can use a variety of different weighting methods that I'll go into. And then more importantly is that we give a variety of different types of t-tests that can establish either difference or similarity, which are different statistical concepts. And I'm going to be going over why saying something is different might not necessarily mean that it's not the same and vice versa. And then, of course, the other thing that I really want to emphasize here is that we can do this orthogonally. So we can use a technique like fluorescence, FTIR, CD, and Raman, put them all in the same software, run that statistical analysis, and then we'll get 4 different pieces of data that all confirm whether our product is the same or whether it's different. So with that, I want to give now information on how exactly is qHOS operating after you've collected whatever type of data that you want, whether that be UV-Vis or CD, FTIR or Raman or fluorescence, and how is it converting it into this easy to understand metric that states whether our products are similar or different to what's out there on the market or what we've produced. So the scheme of qHOS is actually rather quite simple. So the first thing we're going to end up doing is basically what you do for any type of spectroscopic study. We're going to collect spectra. So we'll collect a group of samples. That'll be our sample set. We'll collect at least 3 sample spectra. And then we'll do the same for what we're going to name our reference spectra. So we're going to collect at least 3 of those spectra in order to develop a reference set in this case. What we're going to do with these references now is they could be a variety of things. This could be innovator materials, like I previously mentioned. In particular with MABs, you have innovator materials that go after a specific target, and then maybe someone comes along, finds a better way or a simpler way to produce that material. And that'll be our biosimilar that we'll want to match with whatever the innovator is, because that'll be confirmation that the structures are the same. This could be a case where our references are maybe previous batch scans. So if we're producing these materials in batches and we want to make sure that they're consistently the same, we can take a previous batch, match it up to whatever our new material is, our samples in this case, and this will tell us whether our samples are maybe changing over time. So maybe there's fluctuations in temperature, or maybe the pH is getting a little wonky. And then, of course, we can use something like a NIST reference or a USP reference. So they have a wide number of reference materials that can be really used to develop these reference databases in the qHOS software for comparison. So once we've collected our reference spectra, what we'll go ahead and do is we'll take each of those and we'll develop a mean spectrum of those references, so an average of all of them. And then we'll go through and we'll calculate what's known as the weighted Euclidean distance of each of those reference spectra that we've taken. And we're going to be able to assign them a distance value on this difference plot. So here is the distance of our references. And what we'll go ahead and do is we'll also do that with our samples now. So we take each of these spectra, we go through and compare it with the mean spectrum of the references, calculate a Euclidean distance, and now we can also plot our samples on this distance curve here. In this case, we're going to assume that there's a normal distribution, so a sort of Gaussian normal type distribution of our spectra. And we'll use that to establish a confidence interval for our reference material that basically tells us if it's in this range, it's a match with our reference, which means that the sample is good. In this case, we can see that our sample mean lies far outside of that confidence interval. And so in this case, the t-test is going to give us a failing basically test here, and it's going to tell us that our sample is not identical to our reference. And this is essentially how all of these statistical calculations would work in the qHOS package. All of this is, of course, done on the background, so a user doesn't have to worry about all these statistics. Instead, what they're just presented with is something like this distance curve. And I'll show the actual program interface after a little bit more explanation of what's going on behind the scenes. So I mentioned that we're using what's called a weighted Euclidean distance. And in statistical comparison of spectra, it's really important to get the weighting right. So there's some portions of the spectra that are maybe not important for determining structural information. And weighting really helps identify where the regions are that we should be paying attention to. And so when we do the sample versus reference spectrum comparison at the top, we'll subtract those and then square them. But then we can also apply a weighting function in order to highlight the areas that we deem the most important. And in qHOS, we give 2 options for the type of weighting. We give what's called a noise method and then a spectral change method. The main way that most statistical packages now do weighting is in terms of spectral intensity. But if you're familiar with CD, you know that there are basically points where the signal is 0, right? So you have 0 crossing points that I'll show on the next slide. And if you use spectral intensity weighting, that can become a bit of an issue. And so here, I'm going to outline what the difference between those three methods are and why you might pick one over the other. So on the top here, we have our sample spectrum in blue. If we use our noise weighting function in the middle here, this is our green line. This is based on the light level in the instrument. So for any spectroscopic technique, the way that you're always doing these measurements is you have some light hitting your detector. So if there's low amounts of light hitting the detector, it's going to produce a noisier signal, which means that you'll have much more obviously noise in your data. And that can really impact the statistics when we're doing these types of experiments. And so what the noise weighting spectrum does is it says, if there's not enough light hitting the detector, those regions should be weighted a little bit more fairly because there's a little bit more noise in those signals. But more importantly, in terms of CD, is that if we compare this to the spectral intensity, this sample has a zero crossing point right here, outlined by this red circle. And if we look at the way that a spectral intensity weighting function would weight this region, it basically says that it's not important. And a large number of statistical analysis packages currently on the market will do this type of spectral intensity weighting, which is fine for things like UV-Vis or FTIR, where all of the signals are positive. But in a different spectrum, such as CD, this becomes quickly problematic because these zero crossing points are actually vital information for determining whether a CD spectrum is correct or not. The third type of weighting that we can offer is what's known as spectral change. And in this case, what you're doing is you're comparing a native state sample. You would have a second type of spectrum where you know that the sample is denatured. And effectively what's done here is it weighs more heavily the regions where you know that your spectrum is going to change. So if you know that your spectrum is likely to change as a function of pH change, and you're worried about pH possibly becoming inconsistent as you're doing whatever batch processing you're doing, or if you're trying to develop new formulations, you can use the spectral change weighting function, and it'll more heavily weight the portions of the spectrum that are known to change from denaturization. And so you can more effectively pinpoint when those changes are happening. In addition to offering a variety of weighting functions, we offer a variety of t-tests. But the important thing I want to talk about here is that we offer basically standard t-tests, both student and Welch's. But we offer something also that's called a TOS, or a two one-sided t-test, which is also known as the equivalence t-test. And I want to get into what the difference between those two groups is. And I'm going to talk about that in terms of the Welch's t-test and the two one-sided t-test. So if you've ever ran a t-test calculation, you know that we have to have, like I said before, our samples, and we have to compare them to a reference. And so if we run a set of samples, and then we've established the limitation of where we expect our spectra to be in Euclidean distance based on reference sets, a standard Welch's t-test is asking the question, are these samples statistically different? So if that t-test fails, it indicates that the products are different, right? So in that case, we would say these two structures are most likely not the same. Now, if we pass our t-test, and this is where statistics sort of gets maybe a little confusing with the way it words things, is that the passing means that the products can't be proven to be different, right? So they're not necessarily different statistically, but it doesn't necessarily mean that they're equivalent or they're the same, right? The t-test can just say, statistically, it's likely that these products are not different. Now, with a two one-sided t-test, or the TOS, this is actually asking the question, are the samples the same, right? So if it fails the TOS, basically that means your products are not the same, right? So they're different materials. But in the case of a pass, it's not that the products are quote unquote not different. It is actually showing that the products are equivalent in this case. So the TOS method becomes much stronger statistical evidence that your material is the same. So with that background on how the statistical analysis is really working and the type of options that it offers, I want to get into how the user interface presents data, and a little bit of how data is inserted, but it's fairly simple, so we won't be here too long. The first option that you're presented with is the option of reference creation. So this would be developing a reference data set in order to make statistical comparisons. Here on the left panel, we have the CD spectrum of our reference material. It displays a curve for each one of the trials that we've ran here. And then this gray outline, in this case, is our 2 standard deviation of those spectra. On the right-hand side, it's presenting the Euclidean distance, like I showed previous slides back. And it gives a point for each of the spectra taken. And it also shows where the average spectrum is. One of the other things that's nice that it gives you is it also shows you the frequency of your reference spectrum distance relative to the model that's set up. And this is nice because in a lot of statistical analysis software, sometimes they won't show you this distance curve. And here we can see that the highest frequency point is a little bit left of where the average is according to the confidence interval. And so if we thought maybe we want a little bit more consistent frequency, we could run further references in order to shift this box towards the middle. Or we could say we're sort of fine with this type of deviation. It's not too high, and we could use this as our reference data set. So one of the things that's nice about the software is everything is presented both in a visual nature on the top panel, but then also we provide sort of the spreadsheet view where you can just simply look at distances. So whether you're used to looking at things graphically or in terms of numbers, we offer both of those methods here. After someone's made a reference set in the reference creation, that is saved. And so once you've sort of made your reference material, you can keep on using that reference model as long as you want, or until you need to make a new reference model for a new material. And then we can start actually doing the similarity assessment. And similarity assessment is presented in very much the same way, where we have this graphical panel where now the red is our reference spectrum and the blue is the average of our samples. And then to the right, we're going to give that same distance panel. And so this way we can quickly verify visually how similar things are. So we can clearly see the sample spectrum fits nicely in that standard deviation. We can look at the distance on the right here and see that that also fits nicely in our reference. But then also the qHOS is going to give those hard numbers. So it's going to give us a p-value, a t-value, a Euclidean distance of the average. And then of course it's going to establish a pass or fail. And so we're offering both a visual identification of similarity and something that's more quantitative in line with what the ICH and the FDA require. And so now that we've got a handle of how this software is operating, what it looks like to actually use it, and then why we've developed it the way we are, we're going to dive into an application using the software. And like we previously mentioned, we're going to use Raman, FTIR, and CD, and then we're going to return back to the monoclonal antibodies now. So just to recap, we can have a material known as an innovator that is specifically designed to target a specific site. And then we can develop new materials, biosimilars, which are aimed at matching the structure of those innovator molecules and targeting the same cells, the same types of diseases. Early on in the development process of these materials is a lot of the time where this chemical characterization takes place. And in terms of proteins, there's 2 major types of structure that we're really interested in terms of spectroscopy. The first is secondary structure, which is our alpha helix, beta sheet, turns, and random coils. And those are really nicely identified by either far UV CD or FTIR. And so those methods will work in tandem to identify secondary structure. In addition to secondary structure, we have tertiary structure, which is the final folding stages of the individual units of our protein. And that's best characterized with near UV CD, Raman, or fluorescence spectroscopy. And the combination of these techniques, as I mentioned before, is what we'll call orthogonal assessment. And so in this case, the techniques that we're going to cherry pick to study these monoclonal antibody systems are we're going to use a high throughput CD method that'll look at both secondary and tertiary structure. We're going to use FTIR with an ATR attachment in order to look at secondary structure. And then we're also going to employ the use of Raman microscopy to look at tertiary structure of these monoclonal antibodies. And then we're going to combine all these methods and analyze them in the single qHOS software package. So I've mentioned that we can use qHOS for all of these methods. And what's really nice is that the presentation is exactly the same. It's the same program for each one of these. So for instance, if you teach someone how to run FTIR, you teach them how to analyze the spectra in qHOS, they've now learned how to do that in the CD and Raman spectra as well. So they can easily process these spectra through qHOS after learning it on one of the techniques. In terms of materials, we're going to return back to the rituximab that I mentioned at the very beginning. In particular, we're going to look at two materials. We're going to look at MabTera, which is our innovator, and then we're going to look at Reabni, which is our biosimilar. Both samples are going to be 10 mg per mil, and they're going to have very similar formulation conditions. In addition to these two rituximabs, we're also going to look at trastuzumab. In this case, we're going to be looking at Herceptin, which is an innovator in this field. One of the things I'll note here is that MabTera and Reabni, because they are both a biosimilar and innovator, we expect these to have identical structure, and we'll test that with our 4 different methods that we're using. On the other hand, Herceptin is targeting a different area, so it's going to be an anti-HER2 monoclonal antibody, as opposed to the top 2, which are anti-CD20s. And so we know that Herceptin and MabTera, they're very structurally similar in the tail region, but then at the end of these heads, where the actual binding takes place, Herceptin and MabTera have quite different primary structure and amino acid sequence. And so we'll go through with these techniques and verify whether MabTera and Herceptin have noticeably different structure. Here, I'm also going over materials and methods. I won't dive too deep into the actual settings of these. The main things I want to highlight is, like I said, we're using 4 different methods. So we're doing 2 measurements via CD, FTIR, and Raman. But the really nice thing here is that we're only going to use one analysis method to analyze all 4 data sets here. So we don't have to turn to any different type of statistical analysis package. We can do everything at one PC, essentially. So the first example here is we're going to look at the Herceptin versus the MabTera. So here we can see the near UV tertiary CD spectra. So this is looking at the tertiary structure of our protein. The nice thing about this comparison is that visually we can see there is clear spectral difference. If you've done CD before, you can sort of you have a handle on types of differences you'd expect from 2 spectra. But let's say that you're a new user. And so if you run these 2 samples, it might not be clear to you whether this type of difference is statistically relevant, right? So if we've never ran CD before, the question is, well, this could be just noise, or maybe it's just I'm doing the technique wrong. But we can go through, do the qHOS analysis, and then we can clearly see that if we run multiple of these samples, we can see that the MabTera distance is all the way down here. So this is our reference set. And then if we look at the Herceptin, the distance is way up at around 0.6. And so visually, we'd expect this to fail. And so the qHOS then goes through, it calculates the t-value, p-value. And in this case, it's going to tell us that the similarity assessment quantitatively says these 2 spectra are not the same, right? And that's sort of a really important point to hammer home. We can look at these spectra all day with our eyes and say, well, they're clearly different. But the nice thing about the qHOS is it's really assigning hard numbers to the differences here. That way, when you need to present your data to something like the FDA, if they're doing an audit, you can point here and say, well, the p-value is much below 0.05. And so clearly that means that these two materials are not the same. Next, we're going to be looking at similarity assessment of the secondary structure of these proteins. We're going to be using CD and FTIR. So on the left-hand panel here, this is our secondary structure CD spectrum. And now we're going to be looking at the innovator material MabTera in reference to our biosimilar Reabni. Visually, once again, we can see that they line up quite nicely, that there's a little bit of noise up here towards the 200 nanometer region. And then also we can see that the MabTera has a bit more negative going intensity at the tip there, And so we could ask the question to ourselves, well, will this come out statistically significant? And if we go through and do the qHOS analysis, we find that the Reabni and MabTera line up very nicely on our distance curve. And then the reported p-value is much greater than the significance level of 0.05. And so this will give a similarity rating that passes. Additionally, we can run the same test via ATR FTIR. Here we can see that there's maybe a little bit more difference between the two materials, but we can run it through the same qHOS package, and we can clearly see now that these materials have passed with the p-value and t-value reported here. And so this gives a very comprehensive picture of the secondary structure being consistent between these two. And so based on these two data sets, with the quantitation that was done here, we can clearly show that the two materials are secondarily structurally the same. Now we're going to turn to tertiary structure, once again of Reabni and MabTera, the biosimilar and the innovator molecule. For monoclonal antibodies in particular, tertiary structure is usually what's employed to determine whether two materials are different, because it operates much more like a fingerprint. So from what I've seen personally in the materials that I've ran is that secondary structure of monoclonal antibodies can tend to look the same, but the tertiary structure is really where the differences are highlighted the most. And so here, when we run the two materials, once again, we get that similarity rating has passed for the t-test when we employ near UV CD. And then we can also do the same thing now with Raman. And we can run that through qHOS and it will also give a pass. And here I'd also like to highlight that, so personally, I have some experience with Raman, but not too much. And so if I was looking at this data set, I would sort of not be at odds with determining whether this is significant, because a lot of this looks almost like noise. But it's nice to be able to run this through the qHOS. It sort of gives you these defined values. And then the distance curve here really helps simplify a lot of the complex spectral information that's hidden in this spectrum up here by displaying it in a very simplified manner. And so with that, I'd like to cap off with sort of giving the highlights of the qHOS software again. So in this case, we did statistical analysis for the similarity of monoclonal antibodies. But I really want to also specify that really this can be done with any sample, right? So if you can collect a spectrum of it and you can collect enough, you can develop a reference data set for any type of molecule, including very simple ones. But this software can really use to do statistical analysis of any type of material, not just monoclonal antibodies. I'd like to highlight again, so this includes the TOS test, which is a real equivalency test, as opposed to the t-tests that are typically done, which just go about establishing whether two materials are different or not. And then, of course, there's the orthogonal similarity assessment. So we can employ all of these different spectroscopic methods, which we sell in-house at Jasco, and wrap them all up nicely in this qHOS package, so that once somebody learns how to use one of these instruments, the software is very similar between each of these, so they can get a general feel of the layout. And then the statistical analysis package is exactly the same between all of these instruments. And so once you learn one, you sort of learned how to use any of these techniques, at least from the software end. And then as a sort of last slide, so here we talked about the statistical analysis of monoclonal antibodies, but we have a wide variety of educational resources in addition to this type of thing. So we have a number of different webinars. We've done many on circular dichroism, FTIR, Raman, and VCD. We also have chromatography as well. And those can all be found on our site in case you want to get information either about theory or instrumentation or usage of these instruments. In addition to that, we have a variety of ebooks. We have the knowledge base, which is a repository of literature that's categorized by field, technique, and instrument, which is very easy to go through. And if you're doing, let's say, analysis of monoclonal antibodies and you're using an FTIR, you can check boxes and basically find literature that does exactly what you're looking for. And then, of course, we have robust theory explanation for a wide number of techniques, if that's ever something that you're interested in. And so with that, I'd like to thank you for attending. And if you have any questions, at the top of the bar, I think in Teams, there's a little Q&A. And so you can go ahead and click that box, and that'll give you a little chat type window where you can type out any questions you have, and then we'll field them and answer them. But thank you for listening, and feel free to ask any questions with the Q&A function.
I can see that there is a hand that is raised. If you want, you can type your question in the Q&A.
Yeah, so in terms of picking techniques, usually for me personally, it comes usually down to a matter of concentration. So CD is typically good for lower concentration samples, especially in the terms of monoclonal antibodies. Something like FTIR is more suited to higher concentration samples. So we have things like the penta that can deal with down to 1% concentration of protein, but all the way up to very high end values. And so ATR and FTIR is suited towards higher concentration end. The other thing I think I would consider is that CD, it's a lot more well studied than other techniques, and so there's a lot of background there already, and so it's easier to get started with something like CD than comparison to something like maybe Raman that's a little less dabbled into with protein.
There is another question here. You say, what is your recommended sample size of reference and samples?
Yeah, so the reference set needs at least 3 samples. I mean, what I would recommend is you can operate based on the basically statistical principles. So I would say something like, if you really want to build a robust data set, around 16 references is probably playing it very safe. A lot of the study here, I think we used 8 samples and references. It's either 8 or 10, and that seems to establish a pretty good interval. The other thing to consider is that the more reference samples you run, the tighter your standard deviation is going to get. And so it's going to make it harder for samples to pass if you don't run as many. So what the other thing I would say is you want to make sure that your number of sample and reference spectra are the same when you're doing these types of studies, or potentially having even more sample scans than reference scans.
There is another question here. Say, in comparison of the 2 molecules, can we change the stringency of the cost, and how many replicates can give representative results? And they say they are comparing different lots using CD, but are limited in replicates.
Yeah, that's a good question. So one of the nice things about the way that the software operates is that when you're developing your reference model, if you're using something like a 2 one-sided t-test method, is that it runs through and does the statistical end of things for you. But one of the things you're determining how basically lenient you want to be. So I think by default, it uses 1.5 sigma to establish the tossed safety zone, as I'll call it. But you can change that number to be a bit larger. So if maybe you can run more reference samples than your standard samples, then you can make that tossed window bigger by changing that number. So it's something that you do have a little bit of leeway with. The other thing is that you could also employ something like a Welch's t-test or the student t-test, which will be a lot more lenient also.
Let's see. There is another question. I think I can answer this one. They say, and you confirm, Forrest, they say, can the software be used with data from non-Jasco instruments? And Jasco, for example, Jasco CD, another manufacturer, UBBs, or FTIR instrument?
I know on the reference creation end, there are ways to import CSVs in order to get the files to work, but I would need to double check whether or not that is easy to do, and then if it can be done.
There is another question. It's for the software. You showed data that was in dig? Is this the raw unprocessed data, or how do you keep track of the error propagation in your statistics when performing math or your raw data, such as background, subtraction, and conversion to MRE?
Yes, that's also another good question. So in this case, we're using, we're not converting to MRE. So you could go about this in a number of different ways. So because you're in charge basically with the software of making the reference and the sample data set, let's say you don't want to take a background every time you do this. All you would have to do is for your reference data set, you just leave it no background. And then if you take your samples and the same buffer conditions, the same qVet, then it really doesn't matter that the background's present because it's part of the data comparison at that point. You could run processed background subtracted data in the program as well, but then you'd obviously have to either background subtract before you're taking the spectra or do it after the fact.
There is a question here asking about the cost of the software, but I'm going to invite the attendees today to email us to sales@jascoin.com for questions regarding the price and cost of the software. There is another question here. Is the spectral change weighting provided in the instrument, or it is something a user would provide?
Yeah, so the spectral change weighting, so among the two types we provide the noise weighting, which is simply based on the amount of light hitting the detector, and that's just solely based on the way the instrument is set up. The way the spectral change weighting works is, let's say you're running batches of material and you've known that the pH can sometimes be inconsistent. You could run your material in its native state, have a spectrum for that. You could run it in a purposely denatured state. And then basically when you're setting up your reference, you'll choose the spectral change weighting method, and then you'll give it the spectrum that's associated with your native protein and your denatured protein. And then it'll weight where the change is most highest. So that second one, the spectral change, is determined by the user. So they basically give those two spectra to the software.
There is another question here. I think I can answer that one. It's like the file type is JWS. So JWS is the spectroscopy file type software for Jasco. So yes, the file type is JWS for CD data. I think I'm looking at all the questions. One last question is that you mentioned a test in comparison to a standard t-test. Is there any case where you might use a t-test instead of test? And how necessary is to say something is equivalent with not different?
Yeah, so the comparison between the two methods, it really depends on how stringent you need to be. So I think one of the previous questions was about being limited in terms of replicates. And so in that case, something like the Welch's t-test is probably a more attractive method because it's going to be a lot more lenient. So one of the things about the two one-sided t-tests is that because it is actually establishing sameness or equivalency, it's a lot more strict with what it will accept. But you could always use one of the built-in t-test methods, either student or Welch's, and those will be a lot more lenient with your data. So one of the reasons you might go with that is either if you have low concentration, or if you simply can't do as many replicates, then you can use one of those t-test methods.
I have another question here. Is derivative spectrum a common method in CD spectral analysis?
Yeah, that's a good question. So the CD spectra are different spectra inherently. And so they're the left minus right-handed circularly polarized light absorption. And so those have zero crossing point. And so for those, you're not usually doing the derivative spectrum. You're just leaving the data as is. For something like FTIR, even UV-Vis and Raman, the derivative of the spectrum can sometimes be employed. So if you have, or if anybody's familiar with running samples that maybe have background problems where the spectrum lifts, and so it basically has an offset. If you take the derivative of those spectra, you get rid of that baselining problem. And so sometimes that'll be done to eliminate background issues. The other thing is that by taking the derivative, you're putting a lot more emphasis on the peak position. So at positions where the peak is maximum, it's going to be 0 in your derivative spectrum. And with the noise weighting function that we use, that makes analyzing things like the derivative of either the Raman or the FTIR spectrum viable, which it would normally not be the case.
We have another question here. Is like, is there a function somewhere to give a percentage similarity rather than just pass or fail?
Don't believe there's a metric that it'll spit out that's like percent similarity. Effectively, the distance that's being reported is that. And so if you have your reference data set that has the specified distances for each of those, you can compare those with the Euclidean difference calculated by the samples. And that distance metric effectively operates as how similar is it relative to the reference material. But I don't think by default there's a way to give it as a percentage.
Well, I think I don't see more questions, and maybe with that we can finish our webinar for today. I just want to thank everybody for listening to our software, and again, thank you to Forrest. Thank you very much.
Thank you for everyone for attending. And of course, if you have more questions, feel free to contact us.